All right, so the isolation and identification of Staphylococci is experiment 34. All right, so there's different species of Staphylococci. All right, so Staphylococcus aureus is part of the normal flora, but if it gets in a place that's not supposed to be, it can cause illness with its virulence factors, and this is going to scare you a little bit. Yeah. One of the virulence factors is it produces beta hemolysin. So it's a beta hemolysis. So it does the complete clearing of a blood agar plate. Another one is that it makes the enterotoxin that causes gastroenteritis, which is food poisoning. So it can cause staphylococcal food poisoning and make you sick that way. It produces toxin that causes toxic shock. Produces coagulase, so it can cause the blood clot and what this does, it allows it to wall itself off against the immune system because it's like surrounded by a blood clot instead of and then like the immune system can't get to it to kill it. It causes styles, pimples, boils, and certain wound infections, like staph infection, right? It's got a lot of nosocomial infections. So it's got a lot of it's going to be a result of like surgery, like hospital required infections. And then probably its claim to fame is MRSA, right? Multi-resistant Staphylococcus aureus. It's a bug that is resistant to all of the different antibiotics that we have out there. So if you get a MRSA infection, it's like scary. So we got to like figure out ways to maybe develop some more antibiotics that can help kill Staphylococcus aureus because it's a dangerous little bug. Another one is Staphylococcus epidermidis. It's part of the normal flora as well, and it produces a variety of virulence factors. What we're going to be doing in this experiment is talking. So Staphylococcus just in general is part of the normal microbiota. Like you have epidermidis and aureus on your skin, nose, and throat. They're gram-positive, catalase positive. That occurs in clusters, right? The staph is the clusters. And the thing is they can grow in kind of high salt concentrations, which is probably the reason why it's got food poisoning, because a lot of foods have pretty high salt concentrations, especially in America. And so the fact that it can survive those preservatives is might have to do with why it causes food poisoning in populations. Before we get started on what we did, we have to talk about selective and differential media. So what selective media is media that is designed to promote the growth of specific bacteria and suppress everything else. So if I have a selective medium it's going to specifically grow, let's say E. coli, but it's not going to grow any other types of cells. While differential media says it can grow a variety of different microorganisms, but what it does is it allows you to select within, like it allows you to identify certain characteristics based off of its reaction to the media. So like if you have an E. coli cell and something else, they both can grow in the media, but one is blue when it grows, one's purple when it grows. So you can identify which one's E. coli, which one is not E. coli based off of the color of the colony or something. Or that's just an example. I just made that up. That probably doesn't exist. But anyway, we will be using a selective and differential media in this lab. All right. So we're going to be using mannitol salt agar. All right. So it's selective in the sense that it selects for Staphylococcus bacteria to grow. It's got a very high salt concentration. So everything else kind of dies at that salt concentration, but Staphylococcus continues to grow because it can grow at those high salt concentrations. However, within that Staphylococci kind of classification, it allows you to select for the difference between aureus and epidermidis. All right, and so the way that it does this is through the mannitol salt agar has mannitol in it, which is it's a sugar that you can ferment, and a pH indicator, phenol red. Phenol red is the one that we used earlier, that it starts as red, and then when it gets acidic, it goes to yellow pH. But But then what we also didn't tell you is that when it's basic, it's pink. Okay, so neutral pH phenol red has a red color, but at acidic pH it's yellow, and at basic pH it's pink. Staphylococcus aureus can ferment mannitol, while Staphylococcus epidermidis cannot. So Staphylococcus aureus is going to ferment mannitol and produce the acids that are associated with fermentation. So that means that it's going to grow and it's going to become yellow the more that it ferments. But aureus cannot do that. It's gonna grow in these high salt concentrations. It's gonna just take glucose, probably go through aerobic respiration, and there's going to be red around it because it didn't lower the pH. It's still going to be a neutral pH. So media can be combined, or can be any combination of selective or differential. So you can have something that is neither selective nor differential, and it just, like your nutrient broth is a general, it's just a general growth broth that we use like really early in the semester. Your nutrient broth just to kind of allow stuff to grow. It's not selective or differential. You can have something just be selective in that, you just have something that just allows something to grow on it, and there's no like characteristics that can be differential. You can be just differential, and then you have a general thing that allows everything to grow, but maybe it allows gram positive to grow and be one color, and gram negative to grow and be another color. So you can be just differential, or you can be selective and differential, like we see in this MSA agar, the mannitol salt agar, that selects for Staphylococcus, but then differentiates between epidermidis and aureus. So you can be any combination of selective or differential. It just depends on what the media you're using. Another thing we got to talk about is blood clotting. So so plasma is the liquid portion of the blood. It's like everything that's not the red blood cells. And it contains clotting factors. So fibrinogen is a clotting factor. That's present in the plasma as a soluble protein. So what happens is you have this enzyme coagulase that can cut up fibrinogen and produce fibrin from that fibrinogen. The fibrin will then interact with each other, and that interaction causes like a net of like kind of proteins, I guess you would call it, and like blood cells and all this other stuff, and that net kind of forms a clot. All right. Staphylococcus aureus can produce coagulase, and what that does is it clots blood around the microbes so it can hide from the host defenses. And so what you can do is you can take a swab, put it in a solution, and then add Staphylococcus aureus to it, and you can identify whether or not something is Staphylococcus aureus or not, based off of the property of this production of coagulase. What we did in this lab was we swabbed our noses, right? So swabbing from the environment, and then we played it on the MSA plate and wrapped it in parafilm, and we allowed it to grow. And we examined the plate, and if you have a yellow plate at the end of it, then you probably have Staphylococcus aureus because it underwent fermentation. If it has a red plate at the end of it, then it probably went under. Then it probably means you have Staphylococcus epidermidis from the lack of fermentation products.